Hello, we're glad you've joined us for this live webinar, Androgen Signaling in Advanced Prostate Cancer Models using Non-Invasive Hyperpolarized MRI and Mass Spectrometry. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Agilent Technologies. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. Agilent provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration will provide you with the highest level of confidence in their clinical research solutions. To learn more, visit www.agilent.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Mark Titus, Ph.D., an Associate Professor in the Department of Genitourinary Medical Oncology at the MD Anderson Cancer Center. Dr. Titus earned his Ph.D. from the University of Washington in Seattle. He completed postdoctoral research in the laboratories for reproductive biology at the University of North Carolina School of Medicine at Chapel Hill, North Carolina. His initial faculty appointment was in the Department of Urology at the Roswell Park Cancer Center in Buffalo, New York. In 2012, Dr. Titus joined the faculty in Genitourinary Medical Oncology at MD Anderson. Dr. Titus will now begin his presentation.
Good day to all the attendees. Today I'm going to talk about hyperpolarization MRI and mass spectrometry in metastatic castrate-resistant prostate cancer models. This will be kind of an introduction and an overview of what might happen in the future concerning personalized medicine and how molecular imaging and mass spectrometry information or data will be folded in to this large data set. It may take some time for this to happen, but it might be a very productive way to treat patients in the future. So the outline I'll talk about today is there's an introduction. I'll mention personalized medicine or precision medicine, or as what I'm going to kind of focus on is high definition medicine. I'll introduce molecular imaging. I'll talk about the metastatic prostate cancer models that we use. These are aggressive. The distinction between the two models or model sets is one of them expresses androgen receptor, which is a major driver in prostate cancer, and the other one has lost androgen receptor expression, but it's still a lethal disease. Both diseases are lethal. The phenotype is lethal. Oncologists do a tremendous effort to get people an extension in life, but the diseases that we're focused on are lethal. I'll talk about hyperpolarized MRI, just to introduce the topic, how we collect data on mouse models or the mice hosts. And then I'll talk a little bit about some data that was recently submitted to Nature Biomedical Engineering. The point of this is we're trying to push the envelope and create a data set that will help oncologists predict treatment. It's a little bit in flux right now, but I think it will be interesting in the future. So now I'll introduce high definition medicine. It's this field of personalized medicine to precision medicine, and now it's coming into high definition medicine. it's just a collection of more and more data that has to be analyzed, and then through, just like principal component analysis, you'll be able to pull groups into different treatments or be able to predict lifespan, etc., and maybe even predict prevention treatment. So high-definition medicine is a data-driven practice of medicine that is highly detailed, longitudinal. It's important that you get the longitudinal aspect of this. This may be something that you would be tested right after you're born, at puberty, and then as you go through adulthood. With multiparametric determinants of health, which can, to modify, such as you're going to try to control disease factor risks, control early disease detection. You want to drive precise, precision, and dynamic therapy. You want the therapy to move with the disease so that resistance doesn't occur. And you want to be able to determine preventive intervention. If you're in a specific environment that is conducive to you ending up with leukemia, you want to be able to try to prevent that. It's easier to stop the disease or to prevent the disease than to try to treat it and get rid of it. So high-definition medicine is the biologic and medical science in combination with engineering and computer sciences. This includes bioinformatics. You're bringing a number of disciplines together, interacting them in a cross-disciplinary fashion to give rise to technologies that produce large volumes of clinical information and occasional streams of data that result in large data that require unique capabilities and patient policies. So the federal government and state governments may be involved in this too. The pillars of high-definition medicine: you want to define a personal health baseline. You want high-definition prevention, high-precision treatment that can move as the disease changes. You want your treatment to be able to follow resistance. And then the other goal of this is you want billions, and this is a silly number, but you want large numbers of people so that you have a high-resolution patient and personal profile that will help you do these bioinformatics tests. So the rest of this I'm not going to read. The point of it is that they're trying to define high-definition medicine. They're going to want to define personal baseline, define prevention, define treatment, define high-resolution people. And this is all going to come together and be part of a large, massive data set that will allow oncologists to initiate prevention and personalized or precision therapy. So this is just a pictorial or a cartoon of high-definition medicine, and each of the icons on the left of the slide. The point of this is that mass spectrometry will be involved in this, DNA sequencing will be involved in this, your environment will be measured, your exercise, your diet, the exposures to flu or other diseases in your life. And all of this information will be gathered and put in a high big data system so that your specific profile will be lined up and can be matched to billions of other patients. The idea is that you would have a personal baseline, say, maybe at year 1, at puberty, early adult, late adult, and then as you go through your later life, as you start to present with certain diseases, they have this data already aligned so that they can kind of follow not only your environment, but possibly even your DNA, and be able to assess and try to help predict what will happen and the treatments that will be optimal, as well as prevent certain diseases. And you can see that from the personal baseline to baseline deviations as you move on through your life. So in the end, you may end up with a disease, we'll say cancer of prostate cancer, and you have deep disease characterization because you already know your genome, your environment, and these types of things where you can follow mutations, you can follow methylations or issues that happen with your interaction with the environment, and all of the parameters or all of the icons coming together to try to deliver precision therapy. The point of the precision therapy is that you would be treated to cure, not treated to live with the disease. And that might be something that would be not only cost effective, but it also would be, for the patient, something that's reassuring. If you're treated to cure rather than just treated, and you never really get your quality of life back. So this is an exciting idea, high-definition medicine. So precision oncology, now we've switched from you're being healthy, now you're in trouble and you're going to need some therapy. So it's highly specific targeting of malignant cells or other associated cells, cells in the microenvironment, such as your immune cells. In the tumor microenvironment that help identify cancer lesions and optimize treatments for patient maximum benefit. So that's a twofold point. You want optimum treatment targeted that gives the most benefit to the patient, but you're not over-treating or under-treating. So it's a very precise treatment that maximizes the benefit to the patient as well as cost, to keep the cost down, no over-treatments of patients. Imaging is maybe an essential element of precision oncology that suggests opportunities for targeted cancer screening, staging, treatment, and evaluation of therapeutic benefit. A number of comments. The point is, again, you're trying to get as much information so that patients can be screened, staged, treated with precision, and then an evaluation over time of the benefit. So we're hoping that this will become standard of care possibly in the near future. So cancer biomarkers, imaging at this point, very popular as PET imaging, PET molecular imaging. You can identify site-to-site heterogeneity in metastatic disease. So an example would be, can you distinguish between AR-positive and AR-negative metastatic sites in one patient? You can identify tumor status without biopsy. Biopsies are expensive. Biopsies can cause infection. We would like to eliminate that. We want to predict response to therapy. If you can image and put patients in the correct group for treatment, huge benefit to the clinic and a massive benefit to the patient. So an example of this is in EGFR2 positive breast cancer. They can use an antibody that targets EGFR, and you can take the MRI, and as you see in the images, you may miss a spot by MRI, but you will see it dramatically in the antibody imaging, in the PET molecular imaging. So each imaging modality has its benefits. And what we're trying to do here is we're trying to take PET, CT, and we're going to smash them all together with hyperpolarized imaging, where you will see everything and hopefully see heterogeneity. So another one is, this is F18 fluorine and FDG and FLT PET molecular imaging. Gleevec is a famous treatment. It's a kinase blocker, kinase inhibitor. And what you can see is that in a patient that has, you could follow responsive therapy, you can follow tumor glycolysis in a patient and tumor proliferation if you're using the FLT system. But in section A, you see that GLUT4, which is an importer of glucose into cancer cells. And so the IHC shows that before treatment, you see GLUT4. After treatment, cells that had GLUT4 are no longer there. That's because you targeted them with Gleevec, and those cells have died. So you can monitor how well a specific treatment works, and you can also follow resistance, where cells begin to change and you no longer have GLUT4 expression. So immuno-oncology imaging, this is becoming something that is very interesting, especially in the prostate cancer field. Prostate cancer, there seems to be an exclusion of immune cells, and it's very important, and a major focus or topic of research is to try to understand how to get these immune cells to invade prostate cancer so that these checkpoint inhibitors can be used in combination with other drugs to eliminate prostate cancer and to get the patient to actually fight his own cancer. So one of the problems with FDG PET is that it's misleading due to the metabolic flare that immune cell influx causes. So if you're trying to image a tumor with a sugar and checking PET, you won't see that because cells have a metabolic flare when immune cells come in, and also immune cells are revved up trying to kill a cancer cell, so their metabolic system also increases. Strategies pursued in this are ex vivo labeling of immune cells with image reporter, so you're pulling immune cells out of the patient, putting in a vector, reintroducing the immune cells, and then you can follow them specifically within the tumor. The other one is ex vivo luciferase transfected tumor-infiltrated lymphocytes. It's a bioluminescence assay. Another one is clofarabine, which is a biomarker or PET probe for deoxycytidine kinase. Hematopoietic bone marrow has high cytidine kinase activity, and this might be important when prostate cancer goes to bone, and it's currently not being used that much, but it may be the initiation of a new kind of mechanism to look at prostate cancer in bone and immune infiltrate. So theranostics in prostate cancer is becoming more important now too due to PMSA, but FDG PET is not that useful for imaging prostate, and the reason for this is that the prostate is a very small gland. It's located right below the bladder, where the bladder and ureter connect. It's deep in the pelvis, and its proximity to the bladder means it's an issue because FDG PET, when you give FDG, it immediately is excreted in the bladder. So when you're looking by PET, you end up seeing the bladder, but it obscures the prostate, and you're not getting the best imaging of prostate. You may be able to see something, but it's just like bleaching in a number of, when you're measuring certain things, like in densitometry. If it bleaches out, all of a sudden it kind of disappears. This is the same thing. You get this effect where you see mostly the accumulation of your imaging agent in the bladder and not the prostate, and it doesn't. It's not a very effective way to do this. So an interesting qualifier here is that you can put fluorine 18 on dihydrotestosterone, inject dihydrotestosterone into the patient, and you can watch it accumulate in a metastatic prostate cancer tumor. And then when you give one of these novel or second generation androgen receptor inhibitors like ARN-509, you can watch your image disappear as ARN-509 displaces your labeled DHT, and you start to see your image disappear. So there are ways of using the PET in prostate cancer, but it's more specific, maybe specifically for bone metastatic disease. Theragnostic isotopes, gallium and lutetium, these have been attached to antibodies, and the antibody then will recognize PMSA in metastatic castrate-resistant prostate cancer, somatostatin in neuroendocrine-type tumors, and bombesin in primary prostate cancer. So there are other mechanisms of using theranostic data. You can image, you find out, yes, this is there, and then you can treat accordingly. So multiparametric MRI is a way of doing multiple imaging, but you're using slightly different techniques. There's dynamic contrast enhanced, DCE, MRI, there's MR spectroscopy imaging, diffusion-weighted MRI. And the point of this is that, as you can see in panel A, that's diffusion-weighted MRI. You can pick up disease at where the arrow is, but if you're using another PET imaging agent, which is C11 choline PET, you start to see a little bit better image. And then in a CT scan, the image is there, but it's very difficult to see. So if you combine all of these, you end up with a real nice way of following metastatic prostate cancer. The limitations of MRI is low sensitivity, specificity, patient motion. Also, your GI tract will be moving, and this can cause some issues. It's also very slow, and it really needs to be standardized. So the imaging modality performance and detection of recurrent ovarian cancer, this is another important disease that will be used, or hyperpolarization will be used. So the sensitivity, this is to minimize false negative images, is PET CT is greater than CT, which is about equal to MRI, which is a little bit greater than PET. So if you use PET CT, you're going to get your best sensitivity. Specificity is PET CT greater than PET, greater than CT, which is about equal to MRI. So specificity, minimize false positives, you're after PET CT again. What we're hoping is that hyperpolarization will be able to supersede PET CT, and you'd only have to have one image. So how does hyperpolarized MRI work? So the point of this is that thermally polarization, when you force your nuclei into all the same spin, you never get all of them in the same spin. There's always nuclei that are in the opposite spin. And in hyperpolarized imaging, everybody's in the same spin. You get a massive signal-to-noise increase, and you can see that below panel A, where your thermalized polarization is, there's a lot of background, a lot of noise, a lot of grass. But when you do hyperpolarization, you get this beautiful peak, and it comes way up out of baseline, and it's very hard to miss this, and you can follow this over time. So panel B just shows exactly what's going on when you hyperpolarize C13. You're taking an electron, you push it way up in the polarization ppm, and then it transfers down to carbon, and that boosts carbon back up so that you can see the carbon. So the instrumentation that we used for this study, it's a DNA polarizer. We have a small MR scanner that each mouse host sits in. The tumors are grown on the flank. You can create your hyperpolarized molecule down in the bottom of this DNP polarizer, and you have a transfer molecule along with your C13 molecule. In the sample cup, you have these huge magnets. You blast it with a microwave source, and then you immediately transfer it over and pump it into the animal and start taking your images. So this is how we scan prostate cancer PDXs, patient-derived xenografts in mice. And there's a picture of the clinical instrument, which we have down on South Campus here in MD Anderson. So this is a pictorial, again, or a cartoon of how you can create your hyperpolarized molecule. It's a dissolution dynamic nuclear polarization. So you have enriched C13 contrast agent, which is in blue. This agent helps electrons transfer, and you have the electron paramagnetic agent in green helps electrons transfer to the carbon. So you have high power microwaves at say 1 Kelvin, and magnetic field strength around 3.5 Tesla, and you get the electron to transfer over to the C13. So now you have your hyperpolarized C13 imaging agent. So the non-invasive real-time metabolic imaging that we perform, I'll try to describe that. So it's important that one of the major focuses of cancer, and it's been known for a long time, almost 100 years now, is the Warburg Effect, and it's a part of altered metabolism in cancer cells, where in normal cells you get an anaerobic glycolysis, and you get differentiated tissue will use oxygen. Without oxygen, you'll get glucose to pyruvate to lactate. With oxygen, you'll get glucose to pyruvate. It will enter the mitochondria and go through oxidative phosphorylation. The amount of ATP, of course, everybody knows this, is very high to a very small amount per mole of glucose. But in cancer cells, tumor cells, whether or not there's oxygen, glucose is converted to pyruvate, and most of the pyruvate is converted to lactate. So this makes a tremendous benefit in imaging cancer, because you can hyperpolarize pyruvate and watch it decay to lactate. Whereas the normal cell, most of those epithelial cells are just doing OXPHOS, and there'll be very little lactate there. So you'll be able to image the tumor in the prostate. So this is what we tried to do with our proof of concept in these PDX mice, is we inject the mouse with hyperpolarized pyruvate, and we follow the transition to lactate in the tumor. We're imaging the tumor. So real-time imaging using hyperpolarized MRI, what we have on the right panel is a picture of the NMR collecting data over many transients. The transients are each line as it goes from the bottom to the top of the panel. Each line's probably just a couple seconds. And you can see that the hyperpolarized pyruvate begins to decay over time. Lactate begins to come up and then decay. And you also see pyruvate hydrate, and you see aniline. And on the left hand, you see with the chemistry and the enzymes that are involved in creating these molecules. But you can see that if prostate cancer is taking pyruvate to lactate, the imaging aspect will occur in the tumor, but the surrounding tissue, you won't be able to see that. And you can follow 3 molecules if you have very sensitive equipment: aniline production, lactate production, and bicarbonate production. So what are the benefits of hyperpolarized MRI? Well, it's non-invasive, it's non-radioactive, and it's non-toxic. You're giving a hyperpolarized molecule that the body is already using, so it just uses it like normal pyruvate. Real-time in vivo imaging, you get real-time data from every patient. You can perform experiments faster and potentially cheaper. Now, currently, this system is very expensive, but as this moves forward, just like computer chips, it'll get cheaper and cheaper and cheaper. The critical point here is that it's amenable to longitudinal clinical studies. And going back to high-definition medicine, the longitudinal clinical collection of data is going to be critical in the cure of patients, in trying to get patients all the way through treatment and to a cure. You need the time points before treatment, following treatment, and then at treatment end. So this is not something that's novel. It's been done. This was at UCSF. And what you can see is on the left panel is benign prostate tissue. And you see the hyperpolarized signal come up. It's where pyruvate is. And you can, in the bottom panel, you can see that the lactate, which is the red line, there's not a lot of lactate being produced here, but you see the hyperpolarized pyruvate pulse, but not any transition or metabolism to lactate. However, in prostate cancer, you start to see lactate production. And you can see that in the top right-hand panel where pyruvate peak shows up and the lactate peak shows up. And then the bottom panel, you can see that as pyruvate goes up, lactate starts to increase, and then it tails off with the pyruvate signal. And you can see this image of a human prostate where in the bottom right corner where the two red arrows are, there is prostate cancer. And you're imaging that prostate cancer, but the benign tissue is not showing this transition. So it's a very dynamic, and you can visualize this, and hopefully this will be something that we might be able, this image will be linked to other aspects of the disease, and we're hoping that we can fill in the DNA mutations, deletions, things like that, and help target treatment along with this image. So the prostate cancer models that we're using, first off, this is a kind of a pharmacologic spiral, but you're looking at some of the important drivers of prostate cancer. You have in the initial diagnosis, or at the initial part or the initial growth of prostate cancer, it's a DHT-dependent disease. DHT is the steroid that drives prostate differentiation in development, and it keeps the epithelial cell in its lineage, in its epithelial lineage, and it keeps it doing its job. However, that could kind of go wrong, and you end up with a DHT-dependent cancer. Now, this can then be treated with castration, and then you see these cyclical loops as the tumor begins to change over time. And you start to treat at these arrows. You treat for CYP17, you're treating for androgen receptor, then you treat for SRC kinase. And as the patient goes through these loops, you see the expression of different proteins, and you're trying to treat them according to the expression of these proteins. In the blue box, you see that you get an oncogene activation. Here you have AR-oncogene crosstalk, microenvironment dependence. The tumor is beginning to change. The cancer is beginning to change, and we're hoping that we might be able to image these spirals as we go through this progression of the disease. So you have AR amplification and mutation, paracrine factors. You now have other proteins coming in, FGFR, c-MET, AKT, and these are driving more of an AR-independent disease. You exit from the cycle, and you're finally in an aggressive, more small cell type disease, very deadly. So hopefully we can start imaging and be able to pull these distinct diseases apart and treat specifically so that the patients get the optimum benefit of treatment. So the xenografts that we have here, we have a number of xenografts. The ones that we have used in this study is the 133, the 144, we used the 155, and I think it was the 166. So 133 and the 133 and the 166 have androgen receptor. The 155 and the 144 do not have androgen receptor. So serum PSA in the mice in the 144 and 155, you don't see it. But in the other two PDXs, you do see serum PSA. This just tells you that androgen receptor is driving one disease and it's not in the other. And then some of these diseases will grow and castrate mice, others will not. So here's some relative protein levels in these PDXs. We have the 133, which shows PSMA, CYP17 is expressed, and AR is expressed. Now, as you go to the 144, there's no PMSA because that's driven by androgen receptor. There's no androgen receptor expression. There's no PSMA. You do see a slight amount of CYP17, meaning that these tumors can produce testosterone. However, there's no AR. And in the rest of these tumors, we see some that express smaller amounts of PSA, some under castration resistance or castration do not express PMSA, even though AR is there, because the ligand is not there to drive the AR signaling event. So you can categorize these, and 133, 144, 180/30, and the 163 and the 166 were developed in intact skid male mice. The 133/4 castrate, the 144/13 castrate, the 180/30 castrate are developed in castrate skid mice. So you have to wait for these tumors to grow up after castration. So androgen receptor variant is an important part of the androgen signaling axis, especially in PDXs. You'll see lots of the variant. This usually occurs after castration, and you can see that in these two PDX models, the 133 and the 180. As you see in intact, there's just full-length AR, but in castrate, you start to see some AR-V7 expression. And in the 180/30, the intact, there's a little bit less AR there, but when you castrate, AR comes up and the AR-V7 signal is dramatically increased. That is in the right panel. In the left panel, it just shows the RNA levels, which mirror the Western levels in the right panel. So castration, you think that you may be curing cancer, but the cancer is so dependent on AR that full-length AR is overexpressed and AR-V7 is expressed, and the androgen signaling continues even though you had treatment. So this gives an idea of the expression of AR. And the non-castrated is the left panel. It's just an antibody, an C-terminal antibody that picks up AR in prostate cancer. This is the 133 PDX. And then after castration, you wait 2 or 3 weeks. You can harvest the tumor. And again, you see nuclear localized AR. So the androgen signaling axis is very strong in some of these tumors. And even after treatment with castration, androgen receptor signaling continues. So mass spectrometry now, we measured some ligand levels during the treatment of the 133 PDX. And as you can see, just treating the tumor with vehicle, each tumor will, all of these animals, these mice are castrated, each tumor will make its own androgen. It becomes autonomous. And this is what drives progression of prostate cancer after castration. So you still see 230 nanograms per gram of testosterone in these tumors. And you see dihydrotestosterone there too at about 400 nanograms per gram of tumor. If you treat with an inhibitor of CYP17, which is VT464, this inhibitor blocks the lyase activity of CYP17. CYP17 is a bifunctional enzyme. It has a hydroxylase activity and a lyase activity. The 464 molecule blocks the lyase activity, but not the hydroxylase activity. And then AA underneath it is abiraterone acetate, which is in the clinic right now. But as you can see, the amount of testosterone drops dramatically with each treatment. You get around 60 to 70% inhibition. Dihydrotestosterone also drops dramatically. It drops around 80 to 85%. In the bottom panel, the black line is the vehicle-treated tumor growth over time, and then you have the VT464 in the red line and the AA in the blue line. And you can see that both treatments just slow the tumor growth, but they do not stop the tumor. It's just a suppression of the growth. So another interesting thing is CYP17 inhibition. We do see, and this is by mass spectrometry, a steroid profile that changes dramatically. So in the vehicle-treated mice, cortisol in the tumor is quite high. And then progesterone is also observed in the vehicle tumor. And then when you treat with abiraterone acetate, cortisol starts to disappear. That's because it blocks the hydroxylase activity of CYP17. Whereas progesterone begins to increase, and it's just all over the board depending on each mouse. There's a lot of variability here, but we can still measure progesterone, and it's about the same as the vehicle, although in some cases it can increase dramatically because as CYP17 is inhibited, pregnenolone increases, and then that's converted to progesterone by another steroidogenic enzyme in the tumor. The VT464, the amount of cortisol that is there decreases slightly, but this treatment is designed to allow cortisol production so that you don't have to give prednisone to patients because of your hypothalamus pituitary axis getting off. So it may be used in the future, we'll see. And then the progesterone levels are very similar to vehicle and not quite as dramatically impacted. So here's a hyperpolarization of one of the tumors. This is 180/30. This is an androgen positive tumor. And what you can see is the pyruvate peak there in the middle. This would be the middle panel. It's the middle peak. The transients, the single transients taken every 2 seconds. So from the bottom to the top, you can see pyruvate pulse comes in and then it decays over time. And you can see the lactate peak come up and the aniline and CO3, HCO3 peak come up. But the lactate peak comes up and then disappears, and you can see it on the side. On the right panel, we've just shown one of the transients for each of the tumors that we measured. So the top one is 133. The lactate peak seems to be a little bit bigger. 180/30, lactate's a little lower. The 152, lactate is there, but it's very low. And the 144/13 peak, pyruvate is there, but the lactate peak is very small. On the far left panel is just an example of kind of how the tumor looks according to the imaging, how the MRI sees the tumor, and then we have to tell where the pixels are supposed to be. So you squish it down to where it's just covering the tumor. So hyperpolarized MRI, what we're looking at in these tumors is we're really concentrating on the conversion of pyruvate to lactate. This is accomplished by an enzyme, lactate dehydrogenase. Now this flux through just one enzyme is actually impacted by a number of proteins, from glucose importer all the way through glycolysis till you get to pyruvate and then conversion to lactate. So what you're kind of measuring is this flux of multiple enzymes and transporters. You're not measuring just one enzyme. So you have to take that into consideration, but you do see in the right panel that when you line up the AR-positive tumors, and this is the lactate level that's measured by hyperpolarization, compared to the androgen receptor negative tumors, they are statistically different, and the amount of lactate produced in the AR-positive is a little bit higher than the AR-negative. So what does this imply? This implies that maybe these tumors that are AR-positive get a little boost in glycolysis, whereas the tumors that are AR-negative might have a little bit less glycolysis. So the prostate cancer PDX androgen signaling, you want to, of course, always prove that your tumors that are AR-positive are being driven by testosterone. The tumors that are AR-negative, they may have testosterone, but they don't have the protein. So in the left panel, you see that we measure testosterone by mass spectrometry, and it's around 200 to 250 picograms per gram of tumor. We also measure epitestosterone. The reason for this is that the mouse model will make epitestosterone, so we want to make sure that we separate the isobars, get a good measurement of the actual ligand that's driving AR. Interestingly, 133 expresses androgen receptor, as we discussed before. 144 does not. However, 144 has CYP17, and it does seem to make some testosterone. This is an interesting aspect of these tumors, and it may be a way of the tumor to impact its microenvironment by supplying testosterone. Multiple other cells in the microenvironment of these tumors can respond to androgen receptor signaling. So you may share ligand between endothelial cells and the source of the ligand, the cancer. So we measured a few bioenergy intermediates. We actually measured quite a few, but I've limited it in this talk. But the point of this is to demonstrate that in the left-hand panel, you see more aniline and lactate produced in the 133 androgen receptor positive tumor than you do in the androgen negative tumor, the 144 tumor. You also see more succinate in the lactate, or in the AR-positive tumor than in the AR-negative tumor. And this is by NMR. And then what we did is usually want to try to use two analytical instruments to show the same ratios or the similar ratios of what you're measuring with one analytical instrument. So the NMR is giving us this data, and then we do mass spectrometry, and we see the similar trends where succinate is high in the AR-positive, it's lower in the AR-negative, and lactate is higher in the AR-positive and lower in the AR-negative. So it's important to have this confirmation using two different analytical instruments demonstrating that lactate is produced in the AR-positive tumor at a higher rate than in the AR-negative tumor. So here are some other intermediates, or bioenergy intermediates. And in this panel, what we did is we took the 133 tumor in the intact animal and measured these analytes, and we also measured it in the castrate animal. So you have mice that have the 133 tumor, you castrate one group of mice and you leave the other group of mice intact, and we followed the differences. And interestingly, you see that by NMR, the fumarate levels are the same, lactate is the same, succinate's the same, aniline is slightly different. But regardless of the removal of ligand, the androgen signaling is still there. And we can demonstrate this by NMR and by mass spec so that the trends are the same. You see that in intact animals and castrate animals, the succinate levels are the same, the lactate levels are the same, and the fumarate levels are the same. Now, quantitatively, we can't cross over, but the point of this is to note the trends. The trends are the same, so we have high confidence that what we're measuring is actually what we're seeing in these tumors. Androgen receptor is still driving signaling in these tumors regardless of loss of ligand. So the way we're going to implement this in a clinical trial is we're going to have randomization of 2-to-1. This will be performed by the oncologist in our department. We'll have a baseline image prior to randomization. There'll be an in-house biopsy that's optional. Depending on the patient, some patients will allow biopsies, others won't. We may have biopsies throughout the study. And then it's a 2-to-1 randomization. Group A, abiraterone acetate, ARN-509, cortisol replacement, and chemical castration. In Group B, it will be chemical castration and ARN-509, AR blocker. So we will take these images at baseline 3 weeks, 3 months, and at 6 months. And then after 6 months, the patients will all receive prostatectomies and biopsies will be performed and tissue distributed, and we will be looking at morphology, molecular aspects, protein, steroid, bioenergy. We'll do some RNA, maybe even some RNA-seq. So this is the proposed trial. The workflow that we have is the patients will come in, they'll have hyperpolarized MRI. We'll be doing in parallel PDXs that are from localized high risk, not the PDXs I described, it's another set of PDXs. They'll go through treatment, there'll be prostatectomy, the PDX will be harvested, the clinical data will be collected, NMR data will be collected, LCMS data collected, IHC data. All of this data will be put into bioinformatics, and we'll try to pull apart groups based on principal component analysis and other statistical tests to demonstrate responders versus non-responders. Hopefully this will allow us to link all of the data analysis we have with the hyperpolarized imaging, so in the future, patients coming in being imaged will take some blood, and hopefully we can discriminate and bucket certain people into certain treatments. That's the goal. So this is a large group of people. Multiple other people are involved in this, but GU Med-Onc is led by Dr. Titus. We have other clinicians, myself, and other researchers, including pathology and other cores, all involved in this project. We also have Cancer Systems Imaging. Dr. Pinnica Worms is the lead there. Multiple faculty members involved in this. This is a project that's required a lot of expertise, and we're trying to kind of bring two departments, to a clinical and a hardcore kind of imaging department together and link the information, link the data to improve treatment of prostate cancer patients. I appreciate your time. I'm happy to answer any questions.
Thank you, Dr. Titus, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Titus will answer as many questions as time permits. The first question is, what is the feasibility of high-definition medicine?
So this is more like a thought experiment where high-definition medicine, they're working out details by thinking about it and making proposals. This is not something that at this point is ready to implement. But the idea, I think, is strong, and it's there, and hopefully as this idea is disseminated and people accept it, we can start collecting data, and we will reach billions of patients and have data on that number of patients where you'll have a tremendous statistical analysis to help in getting patients the optimal treatment and benefit.
Hyperpolarized molecules are currently limited, and hyperpolarized MRI is limited to single molecules. So how much information will a hyperpolarized image reveal?
Yeah, so this is a good question, because what we have is this is just pyruvate to lactate transition. But a number of molecules can be hyperpolarized. Succinate is another. But we hope that in the future, chemists will be able to come up and we might be able to derivatize some molecules and hyperpolarize a derivatized molecule that will continue to be cycled through, say, the TCA cycle, or cycled through pentose phosphate pathway, or cycled through nucleic acid metabolism or biosynthesis. And we'll be able to monitor these other pathways in an image by imaging them with hyperpolarized MRI. So you can imagine the possibilities here that you could have a number of imaging events all within the space of maybe an hour for each patient and get a number of interesting data sets that can then be incorporated in high-definition medicine.
Well, I would like to once again thank Dr. Titus for his presentation. Do you have any final comments?
Well, no, so I guess I hope this is kind of an introduction to high-definition medicine, how imaging is progressing in the clinic, and how we're trying to use patient-derived xenografts as a proof of concept that we can actually use hyperpolarized imaging to distinguish patients. And we will continue to work on this, and we hope that in the future the clinical trial and the other PDXs, as we collect data on them, will benefit patients and benefit their therapy.
Thanks again, and I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by email after the webinar. We'd also like to thank our sponsor, Agilent Technologies, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through June 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.